about 5 years ago, we set up a bioimaging mass spectrometry center that was a combination of several of us who were using different techniques, Facundo, Cameron Sellards, and myself, to look at surfaces and the spatial orientation of molecules and biomaterials, but also bringing in people like Tom Orlando, who had ideas about novel ionization methods, and then Mei Wang in biomedical engineering to help us deal with the very large data sets that one obtains when you do this sort of technology. Basically, what we're talking about is a mass spectrometry that goes beyond what you've seen thus far, where you take some material and put it into a vial for an auto sampler or onto a spot for a multi-plate. And we're talking about one where we try to collect data that's in situ in the biological material. And one of the ways this is often depicted in this slide is one in which you take a standard slice of a material obtained with a microtome, like you would if you were getting ready to prepare a microscope slide for a standard staining analysis or to put into a fluorescence microscope. But you put that slide instead onto, or that slice instead onto a multi-plate or onto a special conductive slide that then can be embedded with a multi-material if you're going to be using multi-imaging mass spectrometry, or sprayed with a solvent and method I'll describe in a minute that Facundo uses. If we have multi-matrix material in it, as the plate goes into the instrument, instead of just irradiating it at a specific spot and collecting one spectra, as you've heard about before, spectrum as you've heard about before, you raster across it and collect multiple spectra, literally tens of thousands or more spectra across the surface of the material. And by having instruments that have software that allows you to then work with that data set, you can take the spectra that you collect, decide what ions are of interest, and prepare virtual microscopic images of what the biological material looks like based on that data. So we started off with the Voyager DE star that you've heard about several times before, and basically we're able to do for the first time, not the first time of doing this imaging for brain, but for the first time to image a particular family of molecules in their tissue orientation in this biological sample. Why we were able to do this particular sample and these particular family of molecules was that Cameron Sellards and Yongfang Chen developed a better way of putting the multi-matrix material into the biological sample. The standard way of putting a multi-matrix material in, as you've heard already, is you mix in a solvent, and then that is allowed to crystallize, co-crystallize with the molecules of interest. So if you're going to do this on a surface, you're going to have to spray that material into the biological material. And the problem with the sprayers that were used before that was they didn't create sufficiently small droplets that it was able to go into the biological material without disrupting the structure of the biological material. So by using an oscillating capillary nebulizer to develop that matrix coating, it was possible to get a much better distribution of the matrix material into the biomaterial to see molecules would otherwise be spread around and redistributed and not measured in that way. Then the most recent acquisition for the cores is Bruker Autoflex 3, which has as one of its tremendous features is that it is designed for imaging mass spectrometry. The Voyager was a multi-instrument that was jerry-rigged to do imaging mass spectrometry after the fact, but the Bruker was designed right up front to handle that. And there's a quick little video here that will give you a quick idea about the types of things that are done. Let's see. So Rachel here is, having set the instrument up, turned it on, bringing the multi-plate over that has the microscope slide on it that the tissue samples have been deposited on. Already sprayed the multi-matrix material in. There's another way we can apply it by a sublimation procedure. It goes into the instrument. Then with the software control to orient the plate in the appropriate way, do some calibrations. You heard before in MALDI, you need to calibrate the spot to get the high mass accuracy. Then at this point, we're actually looking at what the microscope slide looks, the whole thing looks like, identifying regions of interest, collecting spectra, then defining which particular ions are of interest and scrolling through those with different color schemes used to differentiate the molecules that you're looking at. And then zooming in a little bit closer, and you'll see a little higher blow up what this could look like, the same scrolling through that group that you just indicated. So you typically have an outline that you sketch from a camera that's on the instrument, which is why we had that pattern around here. You then collect the spectra and display them as you want. And in with this instrument, you do consecutive slices of tissue. So you'll have the one that was right next to this, 8 microns or so away, that you would put on another microscope slide and do whatever characterization of it that you feel is necessary to match what you're seeing with the mass spec imaging with the general view of what the tissue looks like. There are a lot of different methods that one can use for imaging. We just talked about one of them, the MALDI approach. I indicated a moment ago that there are other methods that are used. One is so-called secondary ion mass spectrometry, or SIMS. SIMS can be a very high resolution technique, down into nanometers, if one is using a very high energy ion beam, such as cesium, to activate molecules in a sample and basically blow them off as ions. The problem with that is that this is of such high energy that the molecules of interest to us, biomolecules, are chemically destroyed, and what you're often measuring is a very fundamental molecule like carbon dioxide or ammonia. That's what's left after you've broken it down. So what people usually use if they want to orient something by a SIMS method is a molecule-specific stable isotope, so that they're measuring that stable isotope. There's an emerging method that uses lower energy ion beams, things like charged fullerenes, that are able to bring ions off the surface that are closer to what the parent mass is. The method that Facundo uses extensively, DESI, where you're using desorption electrospray ionization mass spectrometry, you're using a spray of solvent that both releases the sample from the surface and conveys the charge to it so that you can measure the mass spectrum with that. And then there are other variations on a theme. Part of what we've been doing through the Bioimaging Mass Spectrometry Center, as I say, is bringing people together to try to develop new approaches. And Tom Orlando's involvement with Facundo has come up with a microplasma discharge method for developing ions. There are two ways that the samples can be gotten into the mass spectrometer also that differ somewhat fundamentally. One you've heard a lot about, which is using vacuum as a method to bring the ions in, but you can also collect samples with ambient conditions. And Facundo has differentiated these two approaches very nicely in this summary slide, talking about the advantages of the DESI type ambient approach, where you're spraying a solvent onto the sample and releasing molecules off the surface. Very good for in vivo materials that are in their native matrix. And then going all across to what we talked about a moment ago in MALDI or SIMS, where you have to do a lot of sample preparation. You can argue that perhaps if you're not careful, the deposition of the MALDI matrix material could alter the orientation of molecules in the samples. So each of these have their advantages and disadvantages, as Facundo has summarized here. And if you want to know about that in detail, since we're short of time, send me an email and I'll send you the PowerPoint presentation. One of the really valuable things that you have to do if you're going to take this approach is to, as much as possible, use complementary methodologies to extend the information that you get from the study. For example, in studies that Facundo has done with Julia Kabanik here, in looking at the distribution of biomolecules across seaweed, they have the knowledge about the basic chemistry of the molecules, and the question becomes, where are those distributed across the surface? And a DESI type approach is able to add to that. And other methods that are in development in Facundo's lab of things like ion mobility methods for distinguishing molecules that vary by shape rather than their mass characteristics. What I mean by that is you can have molecules that are isomers or isobars that are distinguished by shape rather than by the mass, so that you can get information beyond what you would by a conventional ion separation procedure for identifying and resolving the molecules. An example of what my lab brings, being in biology, is we like to combine other types of omic technologies, such as transcriptomics, to direct our attention to what we want to characterize. That is, to use, for example, a gene expression data set, here expressed in a heat map, meaning that if the genes go up, they are depicted in red, and if they go down, in blue. And so you can see by this map, without even knowing what the items are, that there's an arm that goes red, identifying a family of molecules that were predicted to be elevated in ovarian cancer. And then we use the imaging mass spectrometry to look at a histologic slice from the tumor and identify that indeed in the regions that a histologist would identify as the cancer cells versus the stromal cells, we have a concentration of those molecules that were predicted by the gene data set. And in addition, it's important, because these data sets are so huge, it's not uncommon to have 80,000 spectra across a sample that's about a 1 by 1 millimeter piece. So if you've got that many spectra, how many items? So mass spec imaging, as I hope I've shown you, is on a trajectory to become a standard research tool for taking the power of mass spectrometry to look at molecules and to put them into a biological context of where they are in situ. Indeed, there are companies now that are making instruments that are designed specifically for this purpose, and I want to show you this short video from Shimadzu about a new instrument that they've developed. If I can get the screen in the right place to show you. They call it the IM scope, and although it actually doesn't have capabilities beyond what I've shown you so far, it has an interesting concept that I want to point out to you. The idea now is to get this into a more automated format so that you don't have to have as many trained personnel that handle it. So what they've built into their mass spectrometer is in the instrument a combined microscope and mass spectrometer. So that I mentioned earlier, we take consecutive slices and look at one by microscopy, the other by mass spectrometry. Here they built an instrument that, and when it opens, they open it up in a second. You'll see the microscope component here, the mass spectrometry component here, typical slide just like what we use. The slide is placed into the microscope, collect data from that. And this is only a light microscopy, but you can imagine how, as this is developed further, one could use fluorescence microscopy and more sophisticated techniques. Then that's robotically moved into position in the mass spectrometer so that it's able now to be irradiated by laser light in this little tube that collects the ions that come across the surface to go into the mass spectrometer. So as I say, this gives you a sort of a glimpse into the future of the point where instruments are going to be such that you'll have the sample, you slide it into a tray like you saw Rachel doing a minute ago, but from that point on, the microscopy, possibly optical, possibly fluorescence, there ultimately will probably be no limitation to it, is going to be coupled with the mass spectrometry. So what we have then as we think about this overall is that the characteristics of this is that mass spectrometry is successful when we have well-chosen and designed experiments. That's why I indicated a minute ago that as much as possible, if one can use a powerful technique like genomics to suggest to you what you really want to look for, but there are other ways, of course, you can get to that. Properly prepared samples, as you heard about several times, appropriate equipment, which we have here at Georgia Tech, skilled operators as well we have, and the data analysis tools. We can get data sets that are useful for the kind of studies I've indicated. And then there's really a triumvirate of items here. One is the time that you had to invest to do it. The second is the dollars, because although the instruments are in place, and that has a relatively small cost, they do have lasers that are associated with them, and if one spends a long amount of time because you haven't designed your experiment carefully, replacement of a laser can be $5,000, $10,000, $25,000, and then whatever you always need in research, which is just a bit of luck.